Hello everyone, welcome back to the BioBeavers channel, and I'm Dr. Pragya. We'll be continuing with the same paper as we had been doing. So we completed question number 1 of the Feb March paper 5-2 of Feb March 2024, and we are going to continue with the second question. We've completed the first question, which had the graph question and which had the big 6 marker and the 5 marker of the planning question, which is there. Now we move on to the second question, and the second question is usually based on either the chi-square test, the null hypothesis. A lot of questions are based on genetics in this part. So here it's more of formulas given to you, and you have to kind of provide them with the right working and right answers. So here, when you're attempting the question, it is not just the knowledge of that particular concept which is important, but how you're actually putting down your answers is also very important. So same thing which we followed with question number 1 also. When we have a lot of data which is given here, we have the Drosophila melanogaster small fruit fly data given right here. We will skim through the data. We will make sure we read through the data, underline something which hints to us or we feel is very important at this point of time. So we start with the biologist carried out the investigation. So if you have a look here, the Drosophila melanogaster is a small fruit fly, and wild fruit flies normally have dark red eyes. So since they're talking about the color of the eyes, we know this question might relate to genetics. We underline all the important parts which are here. That is the dark red eyes, and the presence of brown pigment is something you can underline. Then we see that it says what they have done. They've carried out the investigation to determine the roles of 2 genes that are involved in determination. So as I mentioned, because they're talking about eye color, so of course it is related to genetics. And we can see the genetic table right there where they have mentioned what the dominant phenotypes look like, what is the color of these phenotype, brown eyes, and if we have the big R which is there, we have the bright red eyes, and how this is related to the pigment, and the name of the pigments is given right there. Now we move on to the next process. Here they explain the entire experiment. You can just read through it fast. They have some agar jelly containing nutrients and water on day 4. Adult female fruit flies with brown eyes and 4 adult male fruit flies with bright red eyes. So we have the male and the female with different eyes placed together, and we cover it with the cotton wool. So as each point is coming, you should also be thinking at this point, why are they putting the cotton wool? What would be the reason behind it? The specimen tube were left in the warm room for several days. During this time, the parent fruit flies obviously reproduced, and they fed on the agar jelly. On day 7, the parent fruit flies were removed from each specimen tube. Now here another question comes in, why was this done? Then on day 12, the first generation had developed and were ready for crossing to produce the second generation. So let's move on to the first question. As I had been saying, that when we have the diagram coming in and when you're going through that entire procedure, you should start framing and forming your own questions, because that will be really helpful. So the first question which comes in here is, suggest a reason why the biologists removed the parent fruit flies from the specimen tubes on day 7. Okay. So first we put the parent fruit flies, try and understand this experiment. And after they had given birth to the progeny or the younger ones, we remove them. Why would you think that when you have the offspring which have been born, we want to remove the parent fruit flies? Obviously, because at this point, you don't want your parent fruit flies to start reproducing with the new ones. So the answer here is to prevent or to avoid mating between. So the answer is to avoid mating between the parent fruit flies and the progeny. Mating between parent fruit fly and the progeny. Okay. Please remember, I have already said this in my previous video. Please make sure that I am using short forms. You do not use short forms. Also, do not start with the sentence that the biologists removed the parent fruit flies from the specimen tubes because, okay, just start with that one statement of answer which is there. Yes, so we have 1 mark with this question, and we move to the next part. Okay, we move to the next part, and the biologists wanted to cross the first generation with another produce of the second generation. So here we are trying to cross. Earlier we were trying to prevent this. Now the first generation of adult fruit flies in the specimen tube on day 12 were mixed with females and mates and yet had not yet mated. The biologists crossed the first generation of fruit flies with one another. So now at this point, we wanted the first generation to reproduce with the second generation. But when we are putting them together, they are first ensuring that they try and reproduce with each other. So the question which comes in here, which is a 1 marker, asks us, suggest a method that the biologists could use to separate the adult female fruit flies and anesthetize adult male fruit flies and place 4 of each into fresh specimen tube. So first point, they are asking you to separate the female fruit flies as well as the anesthetized adult mate fruit flies. How do you separate these? So the best thing is to look at their eyes. That's the most important thing. How do we look at the eyes of Drosophila? That's the first question which comes in. We cannot just directly have a look at each one of them. So we use a hand lens or a microscope. Okay. We use a hand lens or a microscope. Okay. We use a hand lens or a microscope to be able to differentiate between the male and the female fruit flies. Okay. This is one part. So this was the first part. This is the part which is just before the and. So this is how I've tried to separate. And now you have to place each of the fruit flies. Obviously here, as we remember, as you know, fruit flies around you are very small. If I'm just going to pick up with my hand, it's going to get squished. So we either use forceps or a small brush, maybe a small brush to pick the fruit flies. So you have to clearly write 2 sentences, 2 points. First point, to separate the anesthetized adult female and the male fruit flies, you can use hand lens or microscope. Secondly, to pick them up into the fresh specimen tube, we use forceps or the small brush, which is right there. Helps us to differentiate between the 2 pointers. This was a 1 marker again. So this has actually got a lot of 1 marker, 2 markers. And it's very easy to score in here if you're able to understand. The answers are very simple. And it's just a small extension of small things which you might know. Now we have the proper expected and the observed numbers for each one of them. It says here, the C part which is there, you can see that there's an expected phenotypic ratio and the observed number is this. We have the total number of progeny, and here comes the chi-square test. So the question which comes in here, which is usually very fixed at this point, is the null hypothesis. So always whenever we are stating the null hypothesis, one thing which you have to remember while stating the null hypothesis is that there is no difference between observed and expected, or is there any difference between observed or expected? That's your first question. So we have 9:3:1 ratio. And if you multiply 72 by 9 to get the actual figure, so we have around, so it's more than 9, but around those values is what we are getting. So we see that there is not much difference. So state the null hypothesis. There is not much difference between observed and expected results. There's not much difference between the observed and the expected results. So please, please, please make sure that whenever you're writing the statement, either there's too much difference, but you need to have the terms of observed and expected to be used here when you're talking about each one of them. Okay. And now we move on to the next question. And already we can see the formula which is coming in. We have the chi-square formula which is given to us. Now, how do we start calculating each of these values? So as I clearly pointed out, what are we looking at the values here? So our total progeny number is 1248. That's what we start from. And we have to divide this 1248 multiplied by 9 upon 16. Okay, because the total number is 9 is to 3 is to 3 is to 1. So 9 plus 3, 12, 12 plus 3, 15, 116. So 9 out of 16 will give you a value here, which is 702. This part will be calculated 1248 multiplied by 3 divided by 16. And our value is 234 here. 234, same here. And the last one will be the same number divided by 16, which gives us a value on the calculator of 78. And if you total this up, it is 1248. So as I said, when we are doing these table questions, please make sure each of the values are done. A common mistake is most of us will tend to calculate these values, the expected values, but we will forget to write down the total number because we think we already know about it. But there's an empty box given to you. It has not been colored in gray color. So there is a very, very clear tendency that there might be a mark which is being awarded for what you write here. Now you do your calculations of O minus E. So you do O minus E for each one of them. Fit in the values very clearly. You square them up with the help of a calculator. And then you have the O minus E square divided by E values. So to make your life a little bit easy, what I'll do is I'll actually do each of the calculation. So O minus E square. So when we're talking about the O minus E coming on to first, so we have the 702 minus 691. That's your first value. Okay. 702 minus 691. That gives you a value of 11. Okay. And then we have a so please remember we are doing O minus E. So how do you get your marks? 691 minus this, it's a minus value. Make sure you are writing this minus. Similarly, we have 260 minus 234, which is a 26 right here. It will be a 26 right here. When we do a 72 minus 78, we get a value of minus 6. Okay. Now, when we try and do the squares for each one of them, we get a square. This is 121. All these will be positive values. 26. What is the square of 26? Is what you get as 676. So 676 right here. 676 right here. And the last one is 6 6 are 36. So we've put in our values. Here, O minus E whole square divided by E. So when you do the divisions here, your answer are going to come in points. So obviously the question comes in, till what significant figures do I give? So this is where your answer is. It says 4 significant figures. So we have 0.172, 4 significant figures here, 2.889 when I do the actual calculation. Okay, 0.346. Okay, and then we have 0.462. So if you look at each of these values, there's a small mistake which we did here. So here the two values were different. Make sure you don't do this mistake, which is a very, very common mistake everybody does. Because remember, when we were doing the expected one, we were expecting our values to be the same. Make sure you don't do those mistakes right here. So here we are doing a 225. So we are doing a 225 minus 234. The answer here is not 26. Okay, the answer here is not 26. It is minus 9. And that is why we get a different value here. So we have minus 9 here. If you have a look at each of these values very carefully. Okay. Make sure the mistake which I did, you do not do that, because the O minus E values will be different. The E values will be the same. So when we are looking at the square values, they change too. And we have a value of 81 here. Okay. We have a value of 81 here. And that's why we have different value of O minus E square. Again, as I have said, there's an empty white box here. You need to add each one of them. And again, we should have 4 significant figures right there given to you. Please make sure this table is completed properly in this manner. You can take a screenshot right away. And after this is when you're going to calculate the chi-square, putting everything into the formula which is given to you, and the value comes out to be 3.869, and that is your chi-square value. And that's what you're getting the marks for. So please make sure all your points are there. I pointed out, please do not make this mistake. I made it right here. I suggest this is a common mistake most of the students have been making. Please read carefully. You're not in a hurry. Take your time. Do this question. It's only for 3 marks. And for 3 marks, there's a lot of writing and calculation which has to be done. Okay. So let's move on to the next question. And please make sure you have noted down all the points. Okay. And remember to read the question where it is written 4 significant figures. You should not be giving more than 4 or less than 4 for sure here. And with this, we move to the next question. And make sure you put in the value of chi-square, that is 3.869 right here. The next question is based on the table. And here the question is asking you that using this table which is given to you, explain whether the null hypothesis would be accepted or rejected. So our value for null hypothesis was 3.869. Okay. So our value which comes out from the chi-square calculation which we did was 3.869. Now, how will I say that my null hypothesis is rejected or not? First of all, what are my degrees of freedom? So our degree of freedom was 4 values is what we had. We had the 9:3:3:1 value. So we had 4. So our degree of freedom becomes n minus 1. That is 3. So we are going to be looking at this column, and we are going to be looking at the value of 0.05 to talk about null hypothesis being relevant or not. So first thing is, when we look at, so you clearly point out, first thing is that at degree of freedom 3, so you will write down at degree of freedom 3 and p.05, the value for probability which comes in, the probability value, the critical value, sorry, which comes up according to the table is 7.815. Okay. And our value of 3.869 is quite low than that. Okay. As a result of which, what are we trying to say here? That this is the value which is accepted, above which we will not consider that our null hypothesis is correct. So, but our value for chi-square, which we calculated, is much more lesser for this. So, our null hypothesis is accepted. Please, guys, I know you understand that the null hypothesis has been accepted. And if you give this data, it is presumed that it is accepted. Please do not presume it this way. The null hypothesis has to be accepted, and you have to write that statement very clearly. So first thing is, pick up the degree of freedom, explain what probability value have you taken at 0.05, you looked at 7.815, and our value is much more lesser than that. So obviously our result states that the null hypothesis has been accepted. Okay, so it's a 2 marker. We need 2 points right there. Moving on to the next question, which is an extension of whatever will be given for you in the genetics question. It can either be Spearman coefficient, it can be chi-square test, it can be a simple null hypothesis statement, it can be the Pearson coefficient. But please remember, for most of them, the formulas are given. Also, the only mark which most of you lose is one of the mistakes which I also pointed out right there. Please make sure each of the values are correctly written. They have all the correct significant figures which have been fitted in. Now we move on to the next part, which is an extension. So let's first try and move to the question and then we'll come back. It's more about chromatography. So here they are asking to calculate the RF. So RF is clearly calculated by distance moved by the pigment from distance from origin to the solvent. So here is when you will be picking up your pencil. You'll be picking up your pencil as well as scale. So here, what is the RF value for pigment 1? So the distance moved, okay? First is distance moved by the pigment and distance from origin to the solvent front. So distance moved by the pigment, okay? That's my pigment 1. And distance from origin to the solvent front. So when we are talking about distance from origin and the solvent front, so please make sure the solvent front, as you can see, is right there on the top. So how much distance is there? So obviously your numerator value is going to be there, distance moved by the pigment. You make the measurement from the bottom and you make the measurement from the top, divide the two, and you will be getting a value of 0.73. Please make sure you take a printout of this paper and do it as it is, because when I am trying to do it on the screen, or when we try to do it on the screen, we tend to magnify it, or we tend to change its pixels in a way so that we can see it clearly. So it is always better to take a printout, because your actual board papers are going to be in form of printouts. So please make sure that you take that printout and then attempt this question. This is similar to magnification questions which you had in A levels, AS levels, as well as during your IGCSE times. So now we move on to our next question, and it is the last question. It says state the conclusion that can be made from results of fruit fly breeding in experiment C and from chromatography results. So it's a 4-marker. So 2 marks for giving us the chromatography result and 2 marks for the experiment which was done. Okay. So if we look at the C part, let's go back to the C part. Experiment which is done in the C part very clearly. So here they did a cross and we see that our ratio was quite independent, and we could predict that they have all independently assorted. So first thing that comes as a point made from results in experiment C and from chromatography result is that all of the pigments are separating out clearly. None of them are stuck to each other. So first thing which comes is that our genes are not linked. So the genes for eye color, which we have talked about, the genes for eye color, that is B/b, okay, and R/r are not linked to each other, because we can clearly see a 9:3:3:1 ratio, and clearly indicating that our genes are independently assorting and they're not linked to each other. Okay. This is one point about our experiment. Then the second point which comes out for our experiment, if you clearly try and understand, is here you can talk about how each one of them is related to each one of these proteins. So if we go back, first point is very clear, was very easy to derive. Now we move on to the second part. Here we clearly say that gene B codes for the polypeptide tryptophan carrier protein, and the gene R codes for the guanine protein. Okay. So the experiment C, That is when we saw the blue eyes which are coming in, we can the dark red eyes which are coming in, we can easily say that the fruit flies with the dark red eyes. Okay, the fruit flies with the dark red eyes, the entry of guanine and tryptophan allows pigments to be produced. So brown pigment and both of these pigments are produced. So if we have the dark eye color, it is able to produce both of them. That's why it's giving the dark eye color. So one conclusion comes in from the experiment. Genes are not linked. That was from the table very clearly. Second is very clear. If I have dark gene color and it's written right there in the question, the dark eye color, I have both guanine and tryptophan being expressed and they allow the pigments, both the pigments to be produced. You just have to explain the diagram which is given to you right there. Okay. Now we come to chromatography results. So during our chromatography experiment, if you look very carefully, they say that we can see 4 of the pigments which are in the dark red eyes. So the first point which comes from chromatography, our dark red eyes have all the 4 pigments present in them. So our dark red eyes have all 4 pigments. Dark red eyes have all the 4 pigments according to the chromatography result. And the opposite is clearly, we do not see any of the pigments, so the one with the white eyes have no pigment at all. So here this question appears to be difficult because it's asking you to analyze about 4 pages of data information which is given to you. But all you have to do is go step by step. First was looking at experiment C, predicting a solution about the genes which are there and the dark eye color. Second is looking at the chromatography result and predicting what could actually be there. When you are talking about the experiment in C, you can even talk about how dominant dark red color will have both the dominant genes, and it is clearly visible from the analysis which is given there. Okay. So please make sure when it is a 4 marker, you are able to write 2 and 2 for each one of them. With this, we end the February March 24 5-2 paper, and we have been able to complete it. If you look, the second part was relatively easy to answer. There's a lot of reading which goes into the second part because you have to think according to a lot of data which is given. When you're trying to attempt these questions, if you think that in the starting of the exam for you to think around the paper, it's much more easy. Please make sure you attempt the second question first, because if you think you're more comfortable reading in the starting as compared to the second part, or maybe your brain will get tired when you are towards the end of the examination and you might not be able to read. So please make sure you try and do a few papers where you start from question number 2 instead of question number 1 and devise your own strategy and your own plans to understand that which strategy should you take up. Okay. So with this, I end this video. Please make sure to like my video, and I'll be uploading more papers for you to practice as exams are going to be around the corner. And all the best. Please post your questions if you have any. I'll be happy to answer. Bye bye. Take care.